Chapter 9, the platelet. Platelets are anucleate fragments of the cytoplasm derived from precursors of cells within the bone marrow called megakaryocytes. They circulate in the peripheral blood and live for only 7 to 10 days there. Aged or non-viable platelets are removed by the spleen or the liver. Their main function is to provide wound healing and are the initial responders to vascular endothelial injury. On a Romanowsky stain, they appear small, lavender blue or colorless with reddish purple granules within it. They are 2 to 3 microns normally in shape, and they can be round or oval, but they lack nuclear material. On the smear, you can see that they're fuzzy, small anucleate fragments about 1/5 of the size to 1/2 of the size of a red blood cell. They contain remnants of a Golgi complex, small number of ribosomes, and a small amount of RNA. Now, if the stains are improperly prepared, platelets can appear in small aggregates or clumped together. This appears with neutrophils too, where they can clump around a neutrophil called platelet satellitosis. I've seen many normal small platelets, but there can also be giant platelets or megathrombocytes, which are greater than 5 microns in size. They can be observed in a few disorders if they are seen frequently throughout the peripheral smear. They can be caused from myeloproliferative disorders, or they can just be from a recovery due to severe thrombocytopenia, where the bone marrow is trying to recover and spit out as many platelets as it can into the peripheral circulation. So when we begin our platelet evaluation, we look at our platelet concentration or our platelet count. The normal reference range for an adult is 150 to 400 times 10 to the 9 per liter. 2/3 of these platelets circulate in the peripheral blood. There are 1/3 of platelets that are sequestered in the spleen in case they're needed. So there are always 2 platelet pools in constant equilibrium. Now there's also a reference to a mean platelet volume. This tells us the size of the platelets that we should be seeing in our morphology. So we have our platelet count and our MPV that give us an indication of what we're going to see in the peripheral smear. So when we talk about platelet morphology on our CBC, we have done a platelet estimate. So here, if you're doing a platelet estimate on 100x, within a normal platelet count, you should see 8 to 20 platelets per field of view, which really equates to 1 platelet for every 20 red cells. So you're going to do this estimate to see if it falls within the reference range. When we look at the mean platelet volume now, where the reference interval is 8 to 12 femtoliters, this really tells us the correlation between the size of the platelet. So if you have a very large grouping of platelets, your mean platelet volume will be increased. And you will see that and correlate that on your peripheral smear. And if you see those giant platelets, you will now mark them down on your platelet morphology, saying large platelets or giant platelets present. Now let's discuss Megakaryopoiesis. This is the proliferation and maturation of the precursor megakaryocytes that begin in the bone marrow from the stem cell, which commits to the common myeloid progenitor, which then stems off to the myeloid erythroid progenitor cell. From here, it will differentiate into a mature platelet. So when the precursors are available, they are actively proliferating from the progenitor cell. Platelets are cytoplasmic fragments of mature megakaryocytes. Now, what's very unusual about the megakaryocyte and the formation of platelets is that endocytosis occurs within the cell, meaning they divide within the cell instead of producing daughter cells and increase their DNA ploidy from 8 to 64, and then they can start to pinch off and create fragments or platelets. And each one of these megakaryocytes by the end of stage 4 can produce up to 3,000 platelets. So let's talk about regulation and the cytokine needed to help make platelets. This hormone, thrombopoietin, influences all the stages of platelet production, and it helps in the expansion and renewal of the platelet counts. This hormone is produced in the kidney, liver, and spleen at a constant rate, where with the red cell, EPO is only produced when there is tissue hypoxia. So it's very unusual how TPO binds to the platelets. So within the circulatory system, because TPO is created at a constant rate, it binds to these platelets and is internalized and degraded. So it's not available to stimulate the progenitor cells. So the higher the platelet count in the bone marrow, the more that TPO binds, which decreases the free amount of TPO, which reduces platelet production. The lower the platelet count within the circulation, there is less TPO bound to these platelets, meaning there's more free TPO to go to the bone marrow and stimulate the bone marrow to differentiate into platelets. So megakaryocyte development, this happens in their microenvironment within the bone marrow, and they interact with the bone marrow stromal cells to produce those positive and negative regulators to help develop the growth of these megakaryocytes. They really influence the lineage choice from the myeloid erythroid progenitor to help them develop into the platelet that they need to be. So they have their own vascular niche, and they attach to the endothelial cell lining of the sinusoidal vessels. They are divided into 4 stages of development, which discusses the quantity and characteristics of the cytoplasm, the size, chromatin pattern of the nucleus, and lobulation. Those 4 stages of development are stage 1, Megakaryoblast, stage 2, Promegakaryocyte, stage 3, a granular megakaryocyte, and stage 4, mature megakaryocyte. Within the bone marrow, the Megakaryoblast, or stage 1, is the earliest recognizable cell. It's usually identified by cell surface markers like glycoprotein 2b or 3a or platelet peroxidase. The nuclear maturation occurs first and is complete before the cytoplasm maturation begins. Now remember, the megakaryoblast and cytes, they undergo endomitosis, so they don't actually divide, but their nuclear content divides within the cell. So first you start to see that the cytoplasm increases in volume. The changes are from basophilic non-granular and scant, which is stage 1, to a granular state to an acidic state all the way through stage 4. The nucleus starts to transform from a single round lobe with fine chromatin and visible nucleoli to a lobulated coarse chromatin to no visible nucleoli. And here you see that the nucleus is maturing first due to endomitosis, where the cell's DNA content doubles with no nuclear division, and the cells become polyploid. So the DNA content ranges from 4N to 64N. The Promegakaryocyte, or stage 2, the size now is increasing about 14 to 30 microns in diameter. You start to see an increased cytoplasm, which is primary basophilic. You have few visible eosinophilic cytoplasmic granules starting to form here, and you start to see an indented or bilobed nucleus, which are the beginning of the demarcation membranes that are only visible through electron microscopy. So in stage 3, we start to see, which is the stage on the left or image below, where we start to see the granules starting to form within the cytoplasm. It's 25 to 50 microns in diameter now, where you're seeing numerous cytoplasmic granules with an abundant acidophilic cytoplasm. The nucleus is large or multi-lobed now, and you see no visible nucleoli. Now at stage 4, you see the mature megakaryocyte, or image B, or the last image in the picture below. The cell now is 40 to 100 microns in diameter. You have an abundant acidophilic, very, very granular cytoplasm. And here, we can't see them, but they're starting to make these demarcation zones. And you see no visible nucleoli, and the nucleus is now smaller and very multi-lobed. At this stage, the cell is ready to pinch off those platelets. Now the result is thrombopoiesis, which all occurs within the bone marrow. So as the megakaryocyte matures on the left, showing these different demarcations of how platelets are going to pinch off and form. So you start to see it break apart where the nucleus is extruded and engulfed by a surrounding macrophage. It releases the proplatelets, which are protrusion of the cytoplasm of the megakaryocyte, where it breaks up into individual platelets. And each megakaryocyte can produce 1,000 to 3,000 platelets depending on the nuclear ploidy produced. Now here you can actually see the stages in development where we see the ploidy start to develop and double within the nucleus. Now megakaryocytes at ploidies 8N or higher can undergo that cytoplasmic maturation to produce platelets depending on the need of the body. So platelets are first released between the endothelial cells of the marrow sinuses as proplatelets. Then they break into mature platelets, which are released into the peripheral blood.